Hello and welcome to Chapter 3, Molecules of the Cell. This is lecture by Jeressa Beyer, Part 2. Now we left off talking about polymers and dehydration synthesis. Monomers are linked up to form polymers by dehydration reaction. Basically, you add a monomer onto an elongating chain and one monomer loses a hydroxyl and the other one loses a hydrogen. And you can see this with the short polymer here. Notice multiple monomers linked together, and we're going to add this monomer on. The hydroxyl is right here, the one we're going to lose, and the other one loses the hydrogen, which is right there. When this reaction takes place, water is formed, and you get a longer molecule. Notice you had 1, 2, 3. You have 1, 2, 3, 4 together, which is kind of a simple thing to understand, and it is simple. But you should also know that a dehydration reaction is also known as a condensation reaction. And so equals dehydration. And the way I remember these reactions is I remember what dehydration and condensation means. It means to lose water. Now when you see this, you'll want to pay attention to your textbook website, and here we have it. And we can come down to making and breaking polymers. And we can see a short little short.
Molecules are very large and are built by assembling small molecules, or monomers, into long chains. The resulting molecules are called macromolecules, or polymers. A process of linking monomers, called dehydration synthesis, involves the removal of 2 hydrogen atoms and 1 oxygen atom to form water. One way this might happen is diagrammed here, where several generic monomers are shown with OH groups that could be used for linking. The animation illustrates the joining together of monomers by dehydration.
Now again, that's dehydration synthesis, and that's at your textbook website. So it's a good thing to go watch. And then breaking polymers, a hydrolysis reaction, is the exact opposite. And there are no other names that we're going to use for hydrolysis reactions. Macromolecules can get damaged, or for example, in the case of starch, it's made up of repeating units of glucose. We need to liberate those glucose molecules to use them in the cell. So rather than trash the entire molecule or to break off those individual glucoses, like taking bricks out of a brick wall, the units can be broken off. The monomers can be broken off. You put in the water, break that bond, and you put a hydroxyl on one and a hydrogen on the other. How do you know which way is going to go which? Look, H, OH, OH, H. It's pretty simple. Again, if you go back to the textbook website of that making and breaking polymers, you can see this. And you can review that website and see how they're broken up. The problem with this website is I have no way to start and stop the video. So please go back and review that website. And one thing to notice is that enzymes, you'll see that little blue guy coming along like this. All right. I'm not drawing it right. Let me draw it right. An enzyme comes along and grabs the whole thing and breaks it apart. Not a perfect diagram of an enzyme, but you get the idea. They're biological catalysts. They're neither consumed as part of the reaction. And if you watch that video, there again, 3.3 right here, you can see what's going on. So again, activity, .3, textbook website. Review it to understand hydrolysis, dehydration, condensation reactions. Now macromolecules, again, polymers and monomers. The way to remember this is polymers are the brick wall, and monomers are the bricks. Okay? So brick wall versus the bricks. Now carbohydrates, they're the brick wall. Remember, brick wall. and the monomer are the bricks. Down here is how you can think of that. Carbohydrates function in long-term energy storage and structural components. Lipids are even longer-term energy storage. Proteins control and manufacture structural components, and nucleic acids, information storage and transfer, protein synthesis, and energy transfer. Now really, proteins, this idea of structural components, they make you who you are, okay? And nucleic acids are really just the blueprint for building proteins, okay? That's all nucleic acids really do. They are involved in some energy transfer, namely ATP, adenosine triphosphate, but it's also the blueprint is the main function of these guys. Now again, the polymer or the brick of these guys. All right, carbohydrates, polysaccharide, lipids. Lipids, really, they're a catch-all group. Proteins are called polypeptides. And nucleic acids, of course, are nucleic acids. All right, that's the name of the polymers that you find. And then you have the individual bricks. In other words, that make up these guys. These are linked together just like bricks in a brick wall. Monosaccharides are the bricks that make up polysaccharides. Fatty acids and glycerol are the bricks that make up lipids, but there's also sterols that make them up, because lipids are a catch-all group. You're going to see here in a moment. Anything that can dissolve in oil but not water is considered a lipid. And then we have proteins. They're made of amino acids. And then finally, nucleotides make up nucleic acids. Now I'm going to highlight something here because the words sound similar. Check it out. Amino acids, all right, that's a brick. And of course, it's revolved in proteins. Nucleic acids are a brick wall. Whoops, I hit my thing. And they make up, of course, they're made up of nucleotides. But don't confuse these two, nucleic acids and amino acids. AA, proteins, NA, DNA, nucleotides, okay? Get that terminology straight, because this is the most confusing part of it, of getting these two straight in your head. So be sure and work on that and follow it. Now our first polymer we're going to talk about is carbohydrate. Carbohydrates are a class of molecules. They're composed of carbon, hydrogen, and oxygen, and they're commonly called sugars. They range in size from the small single monomers, the monosaccharides. Recall, monosaccharides are the bricks in our brick wall. Linked monomers, disaccharides, those are two monomers linked together because di means two, and then long polymers, the polysaccharides. Be sure you know that for the test, all right? They function. You need to know their functions for the test. Energy storage, structural components of the cell like cellulose and chitin. In other words, plant cellulose is what gives the strings in celery, for example, and other things that need strength like the seed coat. You'll also see it with a pear, the grittiness, the stone cells of a pear, and chitin, that makes up the exoskeleton. And so the wood of a tree is all cellulose. That's a big polymer out there. Now the monosaccharides. Again, we're talking about the brick. All right. The monosaccharides, the monomers of carbohydrate, are called monosaccharides. And there are several out there that you can find in nature. They're the main fuel source for most cells. They provide raw materials for the manufacture of other molecules. And it usually has a formula that is a multiple of CH2O. Another way of writing that is 1 to 2 to 1. So 1 carbon to 2 hydrogens to 1 oxygen. And again, that's 1 to 2 to 1 right there. They usually have hydroxyl groups and a carbonyl group. Check it out. There's the carbonyl group, and there's the hydroxyls that you see. And the molecule is both an alcohol and an aldehyde or ketone. It has 3 to 7 carbon atoms, and it often has OS in its name. All right. Here's glyceraldehyde. Of course, the exception to the rule to the OS. But notice the carbon is numbered at near the functional group that's double bonded O. And we number the carbons 1, 2, 3. So we have 3 carbons. And check it out. 1, 2, 3, 4, 5, 6. 6 hydrogens to 1, 2, 3 oxygens, that 1 to 2 to 1 ratio. You can reduce that by dividing by 3 into that. Now, the monosaccharides are classified by the number of carbon atoms in the molecule and the arrangement of functional groups. Pentoses have 5 carbons. Check it out, because a pentagon has 5 sides, and hexoses have 6 sides as well as 6 carbons. They're very common. Now, we can also have isomers in the molecule. Notice 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. These are both C6, H12, O6. The same molecular formula, but slightly different arrangement. So check it out. Look at where the oxygen is, and the oxygen is over here. And so that means that OH is in a different place than it is over here. Glucose and fructose. They have slightly different chemical and biological properties. For example, fructose tastes much, much sweeter. This is why it's the sweetener of choice in sodas. You'll use a lot less of it. Now, other forms of carbohydrates, they're usually drawn as a linear model, but in aqueous solutions, monosaccharides fold back on themselves and form ring structures. Most of the time, monosaccharides are drawn in their ring formation. And you can see here the carbons, again, 1, 2, 3, 4, 5, 6, okay, versus, and where the bends are, that's telling you there's 1, 2, 3, 4, 5, and then there's the 5th carbon already drawn for you. But that's what that means. It means those carbons are there wherever a bend in the molecule is. And then we can get very, very simplified and just draw this guy. And this is what you usually see in your book in the diagrams, is that simplified structure. But again, it's a hexagon, 6 sides, glucose. Now, disaccharides. 2 monosaccharides can link together, made by dehydration reaction. Remember, OH and an H is lost. You form water. Glucose and fructose make sucrose, and glucose make maltose. Maltose is found in germinating seeds, and it's not quite as sweet as sucrose. And sucrose is the most common disaccharide. Many plants use it as a fuel source and storage. And if you go to your activities, there's a couple out there that you can see. And this isn't going down like it should. I'm not quite sure why. But there's models of glucose and the activities on carbohydrates. You need to review these out there. Let me see if I can do this. If it's going to work any better. All right, but anyway, go into those models of carbohydrates in your book. Now, why do things taste sweet? Well, you know, it used to mean that fruits were ripe, but it still does. Then fruits were ripe, and so it was the right time to eat them. And it's good, so you went back and got them, because the purpose of an apple is to get you to eat it and then basically poop the seeds out, and that way the tree invades new territories. But another thing is the taste buds on your tongue. We have specific receptors for sweet. It's one of the 4 things that we can taste. What is an artificial sweetener? Basically something made by man, like putting a chloride molecule, a chlorine, onto a sugar and making saccharose is one of them. Now, polysaccharides. More polysaccharides can be added to a disaccharide to form a polysaccharide. And here's one. Notice the glucose units. There's 1, 2, 3, 4, 5, 6. And then you can have branches coming up like this. And of course, this can keep going up if it wants. And another chain can come in. If you can imagine it in your head, with another one branching up here, and then another chain forming that way. And you'll see these can actually get to be very, very, very big. So these molecules are hundreds or thousands of monosaccharide monomers linked together. Again, do you see the idea of bricks in a brick wall with this guy? It's important that you do. All right? And just like a brick wall, it can come up and continue to form. That's kind of a crude drawing, but I think you get the idea now. The two-function polysaccharide, structural components, and long-term energy storage. It's much more efficient to store energy in compact chains versus a lot of individual saccharides, monosaccharides. The two polysaccharides for energy storage, when they're broken apart, glucose is needed. I mean, if you go back and look at this molecule, we can break that glucose off and use it, break that one off. And what's the process to break it off? Did you answer hydrolysis? Good, 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 good. So starch is the polysaccharide found in plants, and glycogen is similar, but it's found in animals. Make sure you know what these guys do, starch and glycogen. Again, you can review it on your textbook website. The two polysaccharides for structure, cellulose, plant cell walls. It's the most abundant organic molecule on Earth, but it can't be digested by animals. So how do termites survive? One answer. We don't have the enzymes to break it down, but bacteria do have the enzymes. The bacteria live in the mouth parts of the termite, or in the gut of the termite, sorry. And they get a nice, happy place to live, and in return, what they pay for rent is breaking down that cellulose and releasing the glucose to the termite. And then, of course, we have chitin out there. It's a polysaccharide used by insects and crustaceans. And we can digest this one. We just don't really want to. And there's what it looks like. Again, check it out. That brick wall right there, there's the individual monomers in our brick wall. Notice it actually forms a helix as it goes along versus glycogen. Notice glycogen is branched. It comes off. There's the branch right there. And cellulose, the last one, wow, that one really does look like bricks in a brick wall, doesn't it? With hydrogen bonds in between, linking this molecule together. And if you pay particular attention, this is what makes all the difference in being able to digest something. Check out the O's in the bond. Pointing down, pointing down, pointing down, we can digest it. Pointing down, pointing down, pointing down. We have enzymes that can break that. But check out cellulose, pointing down, pointing up, pointing down, pointing up. The ones pointing up are the ones we don't have the enzymes for. Bacteria do, but we don't. And lipids are composed largely of carbon and hydrogen, but again, they're not true polymers. They cannot form endless chains of subunits. And we're going to stop with this.